Hey there, everybody. Welcome back to part 3 of our first look at tissues and our study of the epithelial tissues. Today, we're going to go ahead and get started and talk just about some specific types of tissues that we started talking about last time. So last time we looked at the basic layering. So we could have 3 shapes, squamous, cuboidal, or columnar, and we could have 2 layerings, either simple or stratified. And today we're going to kind of look at some of the specific examples of that, where you find them in the body and what they do. So here we have a warm-up question to start you guys off with. So take a second, pause your video, go and answer these 4 questions, and then I'll be back with the answers. All right, so epithelial tissues, why are they avascular? That's a great question. I would say to prevent blood loss is a good answer, but you could write other things like protection maybe, because they are very close to the surface, right? So you don't want to lose them. You don't want to lose blood through your epithelium. Second question says, why must epithelial tissue regenerate quickly? Very good. And the reason why it has to regenerate quickly is because it takes lots of damage. Okay, so it's like your surface. It's like your surface, and it takes a lot of damage. And you probably know this because you've probably damaged your epithelium through, sunburns, scrapes, cuts, that sort of thing. Even inside of your mouth, I'm sure that you've gotten scraped or cut inside your mouth and gotten sore. The third one says, what is the disadvantage of rapid regeneration? And that is, of course, the dreaded cancer. So that's the downside to this. And then finally, why is the basement membrane important? And it kind of acts as like an anchor for epithelium. Basically, it holds it in place, you could say. Something along those lines. It basically holds it down, holds it onto the underlying tissues. Great. All right, moving right along. So today we're going to learn about all the different important types of simple epithelium. The first one is, of course, simple squamous. And so just to go back and review from yesterday, simple squamous was one layer of flattened cells, kind of like this. So we have one squished layer with little flattened nuclei. All right, so simple squamous has a bunch of functions listed here. Everything from diffusion and osmosis, which are very similar. Of course, osmosis is the diffusion of water. But basically, it's so thin that substances can very easily pass. So if you have like a whole bunch of stuff right up out here and very little on the other side, it's very easy for this stuff just to diffuse through a simple squamous epithelium because it's so thin. Same thing with osmosis. Water can move from high to low concentration. Filtration is another one too. So let's say we go to the same example, and let's say we put some like big chunks, like there's some big junk in here. This big junk would not be able to make it through. It would bounce off these cells, but all these little tiny particles could like dissolve through on the other side. So it makes a great filter. And if you think about it, like think about a coffee filter. You have coffee grounds that are pretty large, and then you have water, which has these little tiny molecules. The water goes through the filter, and the coffee stays on the other side. And so that's exactly what we see here. So it's acting as a filter. We can also secrete. So this epithelial tissue can secrete, which means that it can make a product and then release that product to the outside world. So it can secrete like a gland. And it can also do absorption, which I'm going to add an extra cell in here. Absorption is the reverse. So let's say there's a bunch of stuff that's hanging out. It can say, I'm going to absorb all that and put that inside of me. So stuff can pass through. Some things cannot pass through. So it's kind of a selectively permeable membrane here, right? We can release stuff from inside of the cell, or we can absorb stuff into the cell. Very, very flexible tissue. The only downside with simple squamous, it's very thin. So it's not like very strong, if you can imagine that. So there's a couple specific examples of simple squamous. One is called endothelium, and the other one's called mesothelium. And if you look at the prefix here, we have endo, meaning inside, and meso, meaning like middle, right? So the endothelium is the lining of your blood vessels, and actually your heart too. So we're going to say it's the lining of the cardiovascular system. So we have in all of your blood vessels, they have a layer of this endothelium, just like this, and it is allowing little tiny things. For example, think oxygen. Oxygen molecules in your blood can diffuse into the tissues. Big blood cells are not going to leak out through this endothelium. And so your blood stays inside the blood vessel. So it's acting as like a filter. Good. Mesothelium is what I like to call organ skin because it's the outer layer of your organs. Okay. And so what we find out is that, do you remember when we talked about serous membranes? And we talked about how there was like a visceral layer? Well, guess what? That serous membrane is made of mesothelium. So sometimes in anatomy, we have different names for like the same thing. So like if we talk about the whole membrane, it's a serous membrane. If we talk about like what tissue it's made of, it's mesothelium. And you may have heard of like, if you watch enough television, mesothelioma. If you or someone you know has been affected by mesothelioma, call me now, right? Isn't that what they always say? Mesothelioma is a type of cancer that grows in the mesothelium, and it's usually caused by asbestos. So people that have been exposed to asbestos will get this, and literally it's a cancer of the mesothelial covering of the lungs. The more you know. All right, so that's it for simple squamous, our most flexible tissue. Simple cuboidal, if you notice, all of a sudden we go from having 5 different functions of simple squamous down to just 2, secretion and absorption. So if we look at simple cuboidal, we're looking at just one layer of these kind of cube-shaped cells. So notice all of a sudden now we've become thicker. So we have this thicker layer. And now it's not as easy for things to pass through. So like diffusion and osmosis doesn't work very well here. Filtration, it's kind of too thick to be a filter. So this is where these cells are mostly used either for secretion, where they're going to release a substance, or absorption, where they're going to suck a substance up inside of them. So they're kind of like too thick now for stuff to just pass through the other side, is what we're saying. So we've kind of lost some functions, but that's okay. Simple columnar, very similarly, once again, by the way, this is simple cuboidal here. Let's just, not get confused. And then simple columnar, we're going to have one layer of tall cells, like that. Great. So simple columnar, also too thick for us to do any sort of, having things pass through easily. So this was kind of like my analogy about like a coffee filter. Like if you couldn't find a coffee filter, what would you use? You'd probably use like a paper towel because it's close. Like you can absorb and you can filter with all those things. But these are kind of like paper or like cardboard where we're getting like really thick. And so we lose some functions, but that's okay. One of the things that columnar tissues have is they have these things called goblet cells. And that's actually what this guy is right here, is a goblet cell. And I hope you guys know what a goblet is. You've probably heard of one like Harry Potter and the Goblet of Fire. But a goblet is it's a wine glass essentially. So this kind of has the shape of like a wine glass. So it's kind of like fat and then it gets skinny, and you can imagine it's kind of more or less looks like that. Anyway, a goblet cell, once again, looks like that. It's really fat, and then it gets really skinny down at the bottom. And you can imagine, like, if I gave it like a little ring at the bottom, you'd say, hey, it's a wine glass. But what these goblet cells do is they produce mucus. So all these little guys up here are vesicles full of mucus. So we have all these little mucus bubbles that build up and then are released. So if you ever got like a runny nose and your like nose just keeps running forever and you're like, where is all this mucus coming from? And it's driving you crazy. It's coming from goblet cells. And these are found all out throughout like your respiratory tract. So that's why you can get like congestion in your lungs, you can get congestion in your throat, you can get congestion in your nose. So goblets full of mucus. That's really gross, isn't it? Mucus glass. But that's what they do. So basically, these guys produce a ton of mucus, and mucus comes out. And mucus is actually something that we like because it does a lot of different functions, and we'll talk about the functions of mucus later. But basically, mucus is used for 2 things. It's used for lubrication, so it helps things to slide, and it is used for trapping substances. So trapping like dust, bacteria, stuff like that. So imagine like this goblet cell produces like this goopy layer of mucus that covers the other surrounding cells. And now as you breathe in dust, these little dust particles get trapped in here and you get like dusty mucus. And if you've ever breathed in a lot of dust for some reason and you blow your nose, it might come out like some weird color, like gray or black or who knows. That is just really nasty dust-filled mucus. Mucus is also used to lubricate. So for example, like you have mucus that you produce in your esophagus so that when you swallow food, as that food goes down, it slides on this little bit of mucus, kind of like a slug or a snail. All right, another thing that we see in simple columnar is we see microvilli. And microvilli are these finger-like projections that are used for absorption. So basically, here's what we have. So let's say we have this simple columnar cell. And you can see each one of these cells, they're pretty bland, you know, they just have a little bit of surface area. But what if we want to pack more surface area into these cells? Well, what we could do is we could take the top of the cell and just kind of make it like convoluted like this. I kind of made it a little too short, but anyway, you get the idea. But if we start like folding the membrane, we can basically get like this huge sheet of membrane just to pack into this like little tiny area. And that's what microvilli are. So it's kind of like, you know, if you had a piece of paper and someone gave it to you and you had to like store it and you don't have like a folder, you could fold it up and stick it in your pocket. The same thing happens here. So by folding the membrane back and forth, we make these little finger-like projections that stick out the top of our columnar cell. And then this allows us to absorb. So like when substances are going by, they fall down to these little pockets and are absorbed by these. So when we're thinking about absorption of the body, mostly we're thinking about your small intestine. So if you were to look inside of your small intestine, you will see that the entire thing is lined with these microvilli, these finger-like projections on these columnar cells to absorb. We also find that some of these columnar cells can be ciliated. Now, cilia are like this. So here's our columnar cell, and we'll draw another one over here. Cilia are little hair-like extensions, not finger-like, but little hairs. And by hairs, what I mean is that these are like actual folds of the membrane. So the membrane's folded back and forth. Cilia are just like little like hairs that stick out, and they don't have any actual like membrane part to them. And basically what these things do is they move back and forth. They kind of like flow back and forth and are allowing us to move stuff. So here's what happens. Inside of your respiratory tracts, you have some goblet cells that are full of mucus. So there's our mucus goblet. And then next to that, we have some of these ciliated simple columnar cells. And basically what happens is the mucus comes out, and that mucus rides up on top of these cilia and gets pushed away from your lungs. So these cilia all kind of move together, kind of like oars on a boat, and they push this mucus. And so what happens is that this mucus starts out deeper down in your throat, maybe down in your lungs, slowly works its way up, and eventually gets up into your throat, and you go, and you do that little like clearing your throat thing. And when you do that, that knocks this off of the cilia, and you swallow it and break it down. It's pretty gross. You're actually like swallowing your own mucus all the time. But if you guys ever run into someone who's a smoker, and when they cough, instead of being like, it's like, sorry. Basically what happens is when you smoke, tar, which is a component of just about any sort of like combustion of plant material, that tar sticks to the cilia and covers them up and paralyzes them. So now where the cilia used to be used for movement of that mucus, now all of a sudden the mucus can't move. And so smokers have to cough really hard to push it. So cilia is your friend. It's used to move substances, particularly mucus. That's the best example, but there's a couple others that I'm going to show you. All right, now check this out. Ready? There's a picture of a goblet. That's what an actual goblet's supposed to look like. And you can see that kind of looks similar to the goblet cell. And here's another picture of a goblet. This is a guy by the name of Lil John. And Lil John wants you to know that Crunk ain't dead. But with this little thing he's holding here in his hand, this is a goblet. And you can see it's kind of like fat on top, and it's got like a little stem down at the bottom. There you go. Okay. All right. So here's a picture of your endothelium, and you can see it's on the inside of your heart and it's on the inside of your blood vessels. So we just have this very, very thin layer coating this. And this stuff, another thing it does, I forgot to mention, is inside your heart, it's kind of like Teflon in that it allows the blood to slide past because it's very smooth without sticking. So if you didn't have this endothelium, blood would stick to your heart and you would form blood clots, and that would be a bad time. So that's one of the reasons we have endothelium. Mesothelium, like we said, is like your organ skin. It also lines the cavities. And so it makes up the serous membranes, like we said. So in your lungs, it makes the pleura. In the heart, it makes the pericardium. And it makes the peritoneum down in the abdominopelvic cavity. And so there's a good look at it. All right, so here's another picture of simple squamous. So this is some of that peritoneum. And we can see in cross-section, you can see it's like very, very, very thin. Like there's the flattened nucleus. And this is just looking from the side. Top down, it looks kind of like tiles. Here's a simple cuboidal, and we can see that these cells, much more plump nucleus, much more cube-shaped cells. They're not perfectly cubed, but same thing here. There's some nice cube-shaped cells, a nice round nuclei. Very good. Here is some non-ciliated simple columnar. And so these have microvilli. So you can see how it's folded up. This is inside of the small intestine. And you can see there's our friend the goblet cell full of mucus. And basically what this is going to do is this mucus is going to come and like coat the surface to basically help your food slide past. But then all the little nutrients that are broken down from your food get stuck inside these little pockets of microvilli. And that's how it gets absorbed into your bloodstream. So basically we're trying to get the food to move along, but also to absorb some. This is a closer picture, and you can see right here, there's your goblet cell. So this is all your mucus. And you can see this like kind of fringy border. That's just where all those little microvilli are sticking up on the top. All right, and then there's ciliated simple columnar. And you can see in this picture, we once again have the simple columnar cells, so one layer of tall cells. And you can see the individual little cilia. So you can see all the little hairs sticking up here. And this one happens to be inside of a fallopian tube. So remember how I said that substances like mucus are moved? Well, if you notice, there's not a lot of goblet cells. Guess what is being moved down the fallopian tube? That's right, the egg cell, also known as an ovum. So our little ovum is floating down and going down its little path from the ovary all the way down into the uterus on these cilia. So I always think of cilia as kind of like crowd surfing. Like, I don't know if any of you guys have ever crowd surfed, but you know how like everyone like puts their hands up and they just pass the person along. So the ovum actually crowd surfs its way down the fallopian tube. So I always ask people, hey, have you ever crowd surfed before? And they're like, no, I would never do that. And I'm like, actually, you have. And in fact, one of the things that's interesting is that, I don't know if you know this, but fertilization happens in the fallopian tube. So while this little egg cell was going along its happy little way, a little sperm came by, or several, and fertilized that egg. So you were actually fertilized and conceived while crowd surfing. So you can go tell everybody that. All right, and here's another picture. And you can see this one. Here's our cilia. I believe this is respiratory, because you can see there's some goblet cells to make mucus, and there's our little cilia. And so we would just move our mucus all along here. And that's it. So congratulations, you survived yet another lecture. Next time, we're going to talk about all sorts of fun stuff. We're going to talk all about stratified epithelium and go on and learn about glandular epithelium and wrap this thing up. So hopefully we'll see you guys soon.